Hello everyone, welcome to the new session of Microbiology Bacteria. So in this session we are going to study about bacteria, right? And they are part of microbiology because they are the microbes, right? So bacteria comes under the microbes, and that is why they are in we are having a separate branch of biology that is called as microbiology, and we study about all the different types of microbes like bacteria, fungi, algae are also a part of microbe, and also the protozoa, right? So that comes under the microbiology, and in this session we are going to cover up bacteria. So now, what are the things that we are going to cover up in this particular session? So, general characteristic of bacteria. We will see the structure of bacteria, right? Peculiarity of bacteria, how they cellular structure. It's quite different from the other eukaryotic cell, right? Because this bacteria comes under the prokaryotic cell. So that general characteristic we are going to cover up here. And also we are going to study about the reproduction in bacteria. So there are two general types of reproduction that we could see in the case of bacteria, that is asexual reproduction and sexual reproduction. Okay, so that we are going to study here. Okay, and also we will see some common bacterial disease, right? Bacteria causes disease in plants and animals. So like we are studying about the agriculture part, so we will focus basically on the common bacterial disease that occur in the plants. Okay, so let's start the session. Monera is the kingdom of prokaryotes. So how this got name Monera, this kingdom Monera? If we study the 5 kingdom classification, then that constitute 5 different kingdom, that is Monera, okay, then comes the Protista, then comes the fungi and plantae and animalia. This is the 5 kingdom classification. Animalia, right? And in this 5 kingdom classification, here we are having Monera, in which all the prokaryotic cell, the organism that is having prokaryotic cell are placed in this Monera kingdom. Okay, so it includes mostly the early primitive forms of life which are developed from the early stock known as the progenote. So the first organism that has been developed in the sea was a type of prokaryotic cell. In fact, it was not a prokaryotic cell. It is lower grade of organism, that is the primitive form of the prokaryotic cell. Okay, later there are lots of evolution that took place, and finally the prokaryotic cell, the bacterial cell first, that has been formed, and they are called as progenote. Okay, so they developed from the early stock that is known as progenote. So these bacteria has been formed from this progenote, that is the primitive forms of life. Okay, so being the earliest form of life, they are adapted to all the types of habitats. So if we think about the habitat of the time that has been taken place for the evolution of the prokaryotic cell, then at that time you must know that there was no oxygen was there. It means that the atmosphere was reductive type, right? So some of the cells that, now how this prokaryotic cell or how the oxygen has been evolved, because evolution of the oxygen was one of the major steps that took place in the evolution of different organisms, right? So some of the organisms that were present in the sea, they were taking H2S, and they release out sulfur gas. They takes H2S as a nutrient, right? They are sulfur bacteria because they are taking the H2S as a nutrient, and they release out sulfur gas, S2 gas, as a waste. Now what happened in this habitat in which the H2S was present? All the H2S has been used up. Now what is present? H2O. Now accidentally they have taken this H2O in their cell and they released out oxygen instead of sulfur. Now here you can see the structure of H2S and H2O. What is the difference here? It is here in place of sulfur, oxygen is there. So sulfur was released when H2S was taken, S2 gas has been released, and here oxygen is released when they take H2O. Now this environment become oxidative, right? So lots of reaction took place and there has been great change in the bacteria. The cellular organization got changed, right? So all that gives you the next step for the evolution of the organism. Okay, so this was the very, very important step in the evolution of the different types of organism, and the first time this organism has been formed in the sea only, right? So this H2S was present in the sea water. Okay, so being the earliest form of life, they are adapted to all types of habitat. So bacteria are present in fact today also we can see bacteria is present everywhere, right? It is present everywhere. So there are two major groups of monerans, two major groups. So the first one is the archaebacteria, that is also called as the ancient bacteria. It is also called as the ancient bacteria because they can, these bacteria can live in the harsh condition, because earlier the condition of the environment was very harsh. Lot of salt were present, means halophiles. They are also called as halophiles, right? They are present in hot springs, even hot springs. The temperature is very, very high, right? So they are present at those places also, and that is why they are called as archaebacteria, and today also they are found, and that is why they are called as ancient bacteria, because they resembles to the ancient bacteria, because they are having the they are present in the high salt concentrated environment and even in the hot springs. Eubacteria means true. Eu means true. The word eu means true bacteria. So the bacteria that we see, general bacteria that we see today, that is considered as a eubacteria. Okay, and eubacteria is again, it is of further two types, bacteria and cyanobacteria. So the bacteria will have the normal bacterial cell. It will have a cell envelope. It will have no cell bound organelles. It will have center nucleoid, plasmids, like flagella for the locomotion. Now cyanobacteria will have all these things, right? Nucleoid, plasmids, right? Cell envelope. Along with this, they are having chloroplast also. They are having this not chloroplast, chlorophyll. They are present at the lamellae, right? And they do the process of photosynthesis. So you must be knowing that photosynthesis, that require a pigment that is called as chlorophyll. So that chlorophyll is found in this cyanobacteria. So this bacteria and cyanobacteria are considered as true bacteria or eubacteria. Now, what are the archaebacteria? So they are the group of most primitive prokaryote. This is the most primitive type of prokaryote. Why they are called as most primitive type of prokaryote? Because their structure, their physiology all resemble to the primitive type of prokaryote, and which are believed to have evolved immediately after the evolution of the first life. So I've already talked about this, that there are some type of bacteria that were present in the sea, and the condition is very harsh. Okay, so there is an evolution in the bacteria in terms of the nutrient, in terms of the physiology, right? And they have been evolved from those bacteria. So the archaebacteria today, what we see is the most primitive type of prokaryote, and are believed to have evolved immediately after the evolution of the first life. So the first life which was present at that particular time, from those, this archaebacteria has been evolved. So this is very near to the primitive type of organism that has been formed first time on the earth, this archaebacteria. And how we can say this archaebacteria is very close to that, because today also they are found in very harsh type of condition, very harsh type of habitat. They are present in the very harsh type of habitat, as we see in the primitive type of earth environment. Like primitive type of earth environment means temperature, high temperature. They are present in the high temperature, high salinity in water, high salinity. So normal bacteria cannot withstand with these harsh type of condition, but yes, this archaebacteria, they can bear all these condition, and they live as if the most primitive form of bacteria has been present at that particular time. Right? So that is why they are called as archaebacteria. They are characterized by the absence of peptidoglycan in their wall. Means peptidoglycan which is present in the normal bacteria, they do not have such type of peptidoglycan in their wall. Instead, the wall contain protein and non-cellulosic polysaccharides. So the polysaccharide means the carbohydrate part that is non-cellulosic, and they are having a protein that is a different type of, you can say, the chaperonins. Chaperonin are the heat shock protein, that is the protein that helps organism to bear the higher temperature. So this protein is very important, and this chaperonin must not be the type of the chaperonin that is present today bacteria, but yes, it is somewhat like that type of protein that can bear up such high temperature or high salinity. Right? So this is very important from the examination point of view, that this archaebacteria, they are not having the peptidoglycan, normal peptidoglycan that is present in the bacterial cell wall. Instead, the wall contains protein and non-cellulosic polysaccharides. Okay. So they live under the extremely hostile condition where no other organism can dare subside. It means that other organism cannot live in such an hostile condition or such an harsh condition, right? So they live under extremely hostile condition. What are those hostile conditions? Salt pans, salt marsh, hot sulfur springs. Means they are having the very hot water and it is a hot sulfur spring, that is they are having sulfur. there, and they can take those sulfur as a nutrient. They are present in the salt pans. Salt pans mean the salt where the salt are formed, right? And also in the salt marshes. So these are the normal bacteria cannot survive, normal organism cannot survive, but yes, this archaebacteria are found in these hostile condition. Archaebacteria are of three major types. Now you can see there are first one, that is the methogen, the second one, that is the halophiles, and the third one, that is the thermophile. Three different types of archaebacteria are there. So methanogens means the bacteria that is present in the methane. Okay, so methane. So methanogen bacteria is present somewhere in organism also. This you have studied in the lower class as well. So if you go around any cattle like cow or buffalo, right? So what you see, you can smell when you stand just beside to those cattle, you can smell this methane gas. So how this methane gas is coming from their body? Because in their intestine a lot of cellulosic material is there, and these bacteria consume that cellulose and give rise methane, right? This methane, and this bacteria is called as, that is why it is called as methanogenic bacteria or methanogens. So not only there, it is also present in the salt marshes where the lot of organic substances are there. They consume that organic substances and give rise lots of methane. So here also this salt marshes, there also this marshy land, in fact you can say this methanogen bacteria is present. Halophiles are the bacteria that are present in the high salt condition. That is the environment or the habitat that is containing lots of salt. So that is called as halophiles bacteria. Next is the thermoacidophiles. Thermoacidophiles means high temperature and acidic environment. Thermoacidophiles, an acidic environment, the bacteria that is present there. Okay, so they are strict anaerobes. Now we will talk about the methogens. They are strict anaerobes. Anaerobes means they do not take oxygen. They are anaerobe, right? So they do not take oxygen for the you can say the cellular respiration, right? So they undergoes the anaerobic respiration and they give rise a methane as a byproduct. So they occur in the marshy areas where they convert formic acid and carbon dioxide into methane. So they consume this formic acid as well as the carbon dioxide and they give rise this methane gas. This occur in the marshy areas. Okay, and some of the methanogenic archaebacteria live as a symbiont that we have already talked about. That is also called as methanobacterium, that live inside the rumen of the ruminant animals, that is the cow and buffalo. So in their intestine they live, they consume here cellulose, this organic material, and this, when they are present in the marshy areas, they consume formic acid and carbon dioxide and give rise methane. Okay, yeah. So these archaebacteria are helpful to the ruminants in fermentation of cellulose. So they are consuming those cellulose, right? And because the cellulose is not digested by not even by the human, not even by the cattle, but yes, inside the cattle, due to presence of this methanobacterium, it has been digested and methane gas has been released, right? So this is about the methanogen. Now mycoplasma Mycoplasma, that also a part of the bacteria only, right? Mycoplasma, PPLO, pneumonia like organism, right? Sometime full form of this can be asked, that is the pleuropneumonia like organism. So the mycoplasma or the molecules are the simplest and the smallest of the free living prokaryote. These are the simplest type of bacteria, or you can say the living prokaryotes. Okay, so like other prokaryotes, they possesses one envelope, cell wall is absent, they lack organized nucleus and cell organelle. So here if you see, lipoprotein membrane is there because no cell wall is there. Cell wall is totally absent, right? So their envelope is made up of this lipoprotein membrane. And this is one of the peculiarity of this mycoplasma, that it can change its shape. It can change its shape like this when it is moving, right? Like this. It can change its shape, and that is why it is also called as joker of the plant kingdom. Right? So yeah, this is the joker of the plant kingdom. They lack organized nucleus. So no nucleus, as it is in the prokaryote, kept in the prokaryote. So no well organized nucleus is present. Instead, the content of the nucleus is present itself in the cytoplasm. And you can see here this central chromosome is present in the center only, in this cytoplasm only, and that is called as nucleoid, right? As we can see in the case of bacteria, normal bacteria. So here also no membrane bound nucleus is present and no cell organelles is present. So this is the one of the simplest and smallest type of free living prokaryotes. So they are having soluble proteins in their cytoplasm, they are having the ribosomes, right? They are having the DNA content and soluble RNA. So all these things are present in the cytoplasm, but no cell bound organelles we can see in the case of mycoplasma. So they are the heterotrophic in nutrition. They cannot make their own food. They take food from the outer environment, and in fact they release out some of the enzyme. The enzyme digests the organic matter which is present outside their body, and they again take that solubilized and partially digested organic material, right? So some live as a saprophytes, but majority of them are parasites to the plants and animals. So basically they are two types, a saprophytic also and parasitic also. So they are present as a parasite on the plants and animals, and sometimes they live as a saprophyte, means they are present on the dead and decaying matter, and they take all the nutrient that is present there. And how they take the nutrient matter? They release out enzymes. That enzyme digests the organic matter, and that organic matter is again taken by the cell. So this is about the mycoplasma. Halophiles. They are found in the salt-rich substrata like salt pan, salt beds, and the salt marshes. So these are the salt-rich substrata. Salt-rich substrata means the substratum that is having high content of the salt. And what is the salt pan? The salt pan are the place where the salt are made commercially. So there these halophiles bacteria are present. So that is such a harsh condition. No normal bacteria there that cannot survive. Because why they cannot survive? Because the condition of the outer environment is salt. Lot of salt is there. So it take out the water from the bacteria, and bacteria can die, right? But here the halophiles are adapted to such a harsh condition. They do not take out water from their cellular, cellular cell, right? And salt beds also contain all those salt, high concentration of salt, and even the salt marshes. So some of the examples that are the halophiles are the halobacterium and halococcus. They are able to live under the high salt condition due to of 4 reasons. Now this is going to be very important. What are the 4 different reasons due to that halophiles can survive in such a high salt rich condition? Presence of special lipid in the cell membrane. So I told you that they are having a very special type of membrane that do not allow the water to come out from the cell, as they are present in a very high salt concentration habitat. But still, normally what happened due to the process of osmosis, water move from high water condition to the lower condition. Now outside the cell there is having a lot of salt. It means that the condition of the outer environment that is having very less concentration of water, so water will move from inside to outside. But here it do not happen due to the presence of special lipid in the membrane, so it do not allow to happen a normal osmosis process. Occurrence of mucilage covering. It is having a mucilage covering that protect the cell from the high salt condition. Mucilage covering that do not give any type of you can say the effect of those high salt concentration on the cell due to the presence of this mucilage covering. So it provides some sort of protection. Now if you think about the digestion in the stomach, so there is a release of gastric juice. What is present in that? Pepsin enzyme, hydrochloric acid, and mucilage. Now this mucilage that cover ups inside the wall of the stomach, and it provide protection from the high acidic condition, that is the HCl, presence of HCl. In the same way, this mucilage is also provide protection to them. Absence of sap vacuoles. Sap vacuoles are totally absent here. High internal salt content, and they're also having the high internal salt content. So water cannot move outside because inside also they are having the high salt concentration, right? Next is the thermoacidophiles. Archaebacteria have a dual ability to tolerate high temperature as well as high acidity. So that is why they are having thermo, and they are also called as acidophiles. Philes means the type of organism that can be present in the high temperature environment as well as high acidity environment. So that is called as thermoacidophile. So this is also a part of the archaebacteria. We all know this, right? In the classification of the archaebacteria, they are having thermoacidophiles, halophiles, right? Methanogen bacteria, and also that's mycoplasma is there. They often live in the geothermal vent as well as hot sulfur springs. So this geothermal vent means the land is having a thermal vent, means opening through which a high temperature water is coming out, that is around 80 degrees centigrade, and the pH, sometimes the temperature is about 80 degrees centigrade and the pH is around 2, that is highly acidic. And the bacteria that are examples of the thermoacidophiles are thermoplasma and thermoproteus. These are the 2 examples, right? So due to the following reason, they are able to live under the high temperature condition and high acidic condition. What are that? Branch chain lipid in the cell membrane. In the cell membrane, the lipid membrane is quite branched type. The lipid membrane is quite branch type, and the presence of special resistant enzyme. They are having a special type of resistant enzyme that can provide resistance against the high temperature as well as the high acidity that is present outside the cell, right? So this is very, very important. You must understand and you must remember even that why this thermoacidophiles are having what the different type of composition in their membrane. In case of halophiles also, they are having a different composition in their cell membrane, and here also the cell membrane is quite different from the normal cell membrane that is present in the normal bacteria. Now comes the cyanobacteria. So the blue-green algae that is present in the freshwater, seawater, salt marshes, tree trunk and the frozen water. So all these places we can see this blue-green algae, they are present, right? And this blue-green algae are they can do the process of photosynthesis because they are having chlorophyll, right? And where they are found in the fresh water, means that water that do not contain salt in it. Seawater means they are having high salt concentration, so they are present in the seawater as well. Salt marshes, the marshes that are having lot of salt concentration, so there also these cyanobacteria are present. They are present on the bark of the old wooden bark of the tree trunk. So sometime what happened at the hilly region, they are having lots of rainfall, and this is tree trunk is quite moist, right? So these cyanobacteria, you can see they are present at those places also. And in frozen water also, sometimes the water get frozen, right? And in that frozen water, these cyanobacteria are present. So this is one of the very peculiar thing about the cyanobacteria. They are photosynthetic prokaryotes, means the prokaryotic cell, they do not contain chloroplast, but yes, they do contain chlorophyll, right? In which food is stored in the form of cyanophycean starch, lipid globules and protein granules. So the food material means they can make their own food. They can do the process of photosynthesis because they do contain chlorophyll, because some lamellae like structures are there, this membranous lamellae on which the chlorophylls are attached. Now this chlorophyll are helpful in doing the process of photosynthesis, right? And in this photosynthesis, they store cyanophycean starch, they store the food material in the form of lipid globules and also the protein granules. So here, this is one of the example that is called as nostoc. You can see here the nostoc. Okay, now these are 1 cell, 2 cell, 3 cell, means they are present in the chain of a cell, and above that a layer of mucilage, you can see that blue color, right? That is called as the mucilage sheath. Now some of the cell are quite modified, they contain heterocyst. Now this heterocyst is responsible for the nitrogen fixation. So they do the process of nitrogen fixation and they can synthesize protein as well. So that protein is present in as a protein granules. Okay, so this is the very, very important. Cyanobacteria, also referred to as blue-green algae, they have chlorophyll as a similar to the green plants and are photosynthetic autotrophs. Okay, so this cyanobacteria, they are also called as a blue-green algae and they contain chlorophyll similar to the green plants. So we can normally chlorophyll A and chlorophyll B is present in the normal green plant. So that is also present in the chlorophyll, type of chlorophyll that is present in the cyanobacteria, and they are also present in the photosynthetic autotrophs. So cell structure is typically a prokaryotic type, means no cellular organelle is present. Cellular membrane bound organelle is not present, and nucleus is also not present. Instead of nucleus, they are having nucleoid. Okay, Some of these organisms can fix atmospheric nitrogen in the specialized cells that is called as heterocysts. This is very important point from the examination point of view. Like in the cyanobacteria, heterocysts are present, that is nostoc and anabina. These are the two examples of the cyanobacteria. Okay, carbohydrate reserve food material is a special starch known as cyanophycean starch, right? Cyanophycean starch. Cyanophycean starch is a type of a starch in which the carbohydrate reserve food material is present, right? And that is why it is called as from cyanobacteria, cyanophycean starch came this word. Some examples of the cyanobacteria are the nostoc, anabina, oscillatoria, spirulina. These are some of the examples of the cyanobacteria. Okay, coming to the normal bacterial cell, structure of the bacterial cell. What we see here, in shape, bacterial cell are of many types. So here you can see in the round shape, small shape. So that is called as cocci. They are in a rod shaped, and that is why they are called the bacilli. And here you can see some spore like structure. So spores are present in the bacilli, rod shaped bacteria, endospores. Spirilla, spiral type, like spiral type, and they are having the locomotory organ that is called as flagella. They are sometimes in the comma shaped also, right? So in shape, bacterial cell are of, that is why it is called as many types of shape of the bacterial cell we can see. Coccus, spherical or ovoid in outline. So here this is the example, that is cocci. That is, they are spherical in shape or sometimes ovoid, oval. Bacillus, straight or cylindrical rod shaped bacteria. So the bacillus, lactobacillus is a very common type of bacteria that is present in the curd, like lactobacillus. Streptococcus, that is the round shape, and lactobacillus, that is the rod shape. Okay, spirillum, bacterium is coiled like a corkscrew, so spirillum, right? Vibrio, bacterium is like comma or curved rod, like Vibrio cholerae, right? So this is some of the structure. You can see there are a different cellular shape of the bacteria, and according to their shape they got their name also. Okay, here we can see a normal bacterial cell. What we see here, we can see, if we start from the outer side, then we can see a capsule, we can see plasma membrane, we can see the cell wall, and this all unitedly forms cell envelope. Okay, now this plasma membrane which is present inside this this is the innermost, it invaginate at some places and it forms a mesosome like structure. It is a very important for this bacterial cell. Non-membranous structure, that is ribosomes, they are also present here, the purple color you can see, and it is of 70S type. You all know that what is the different types of a ribosome that is present in the bacterial cell and the eukaryotic cell. 70S type and 80S type. 70S type is present in the prokaryotic cell, and 80S that is present in the eukaryotic cell. So this bacteria is a prokaryotic cell. So this bacteria will have 70S type of ribosomes. Cytoplasm is there, and inside the cytoplasm they are having a single chromosome, circular single chromosome like this. Okay, and no membrane is bound, and that is why it is called as nucleoid, a primitive type of nucleus. No membrane bound nucleus is there. In the cytoplasm, we can see lot of plasmid, circular plasmid like this is present. Plasmid is present. Okay, so this is very, very important. And as a locomotory organ, you can see lot of flagella are attached to its surface, and many pili-like structures are also we can see here, right? So we will see one by one what are these structure, why it is present, what is the peculiarity about it. So component of the bacterial cell, starting from the cell envelope. The bacterial cell consists of the cell envelope, cytoplasm, nucleoid, plasmids, inclusion bodies, flagella, pili, and fimbriae. So all these we are going to study. Cell envelope. Cell envelope consists of three components: glycocalyx, cell wall, and cell membrane, right? So this cell envelope consists of the three components, that is the glycocalyx that is present outside the surface, cell wall that is you can say that always present and in the bacteria, and also the cell membrane. But this glycocalyx is modified from bacteria to bacteria. This glycocalyx is also called as the mucilage sheath. Outermost mucilage layer of the cell consists of non-cellulosic polysaccharide with or without protein. So this non-cellulosic polysaccharide means they do not contain any cellulose, right? But they are having the polysaccharide and without any protein, no protein is there. Okay, so it may occur in the form of slime layer, loose sheath like structure, and this slime layer provide a slippery, you can say the slippery texture of the bacteria, and sometimes they are having very hard thick covering that is called as capsule. So this is thick and tough mucilage covering when they are present in a very harsh condition. So they are having a capsule, and the one that is present in the normal form, and they provide attachment to the substratum. So they are having this slime layer that is called as mucilage. Okay, it provides sticky character to the cell. Okay, though this slime layer, it provide very sticky character to the cell. So whatever the substratum is there, it get attached to it. Cell envelope in bacterial cell, that is cell wall. Cell wall lies between the plasma membrane and the glycocalyx, right? The outermost is the glycocalyx and the innermost, means if we move from the outer to the inner side, right? This you must remember, outer and inner. Outer, that is glycocalyx. Glycocalyx is present, right? Then comes the cell wall, and then comes the plasma membrane or the cellular membrane, right? Okay, now this glycocalyx can be of two types: slime layer and capsule. Two types of there. So in a bacteria, either one of that is present, either it is in a slime layer form or either it is in a capsular, hard covering capsular form. Now the cell wall is made up of polysaccharide and amino acid. Very, very important. NAM and NAG. N-acetyl-muramic acid, okay, and N-acetyl glucosamine, NAG and NAM. So the peptidoglycan makes up the rigid protein of the cell wall is called murine. So this is murine, that is the protein component of the cell wall. and it is made up of N-acetyl-glucosamine and N-acetyl-muramic acid. N-acetyl-glucosamine, that is NAG, and N-acetyl-muramic acid, that is NAM. So just remember this name. This is the protein component, and that is present in the cell wall. Very important feature of the bacteria, that their cell wall is having such a arrangement of the protein. Normally, this is also a non-cellulosic. It is a peptidoglycan, means the glycan that is having a protein component attached to it. Now, this protein component is of two types, NAG and NAM. This is not present in the normal cell wall of the plants, and that is why these bacteria can live in a very harsh condition, right? Cell envelope in bacterial cell plasma membrane. Third component after the glycocalyx, after the cell wall, now comes the plasma membrane. Bacterial plasma membrane is of typical membrane. The typical membrane that is present in the eukaryotic also. That is, it is having the lipid bilayer, and protein part. Now this protein part, that is of two type of protein, intrinsic and extrinsic. Intrinsic and extrinsic. Now if you see the cell lipid bilayer like this, it is present. It is having the two poles, hydrophilic and hydrophobic, right? So they are having protein in between. Again, presence of the lipid layer, and sometimes they are having like this extrinsic protein. Extrinsic protein means the protein that is present just to the outer surface of the plasma membrane. It is not traversed between the two bilayer. Not two, that is a single bilayer. So this is, we can say, the lipid bilayer, right? This is the hydrophilic, and this is the hydrophobic. So the one that is present away from the water, that is called as hydrophobic end or the tail, and this is hydrophobic head or the end that is present towards the water, outside of the membrane. And in between these bilayers, sometimes there is a protein that is called as intrinsic protein. This is outside only present, that is called as extrinsic protein. Okay, so it is made up of the phospholipid bilayer. So what type of lipid is there? Phospholipid. Very important. Phospholipid bilayer with various protein, intrinsic, extrinsic and transmembrane. Sometimes there are transmembrane protein that is present here at this level. So that is why it is called as transmembrane protein. So this is the typical type of membrane that is present in the normal bacterial cell also. Okay, so this is all about this session. We will have one more session to complete the cell later part of the cell component of the bacteria, that we will cover up in the next session. Thank you very much.